Hi there, my name is Gil Cohen, part of the Cella Laboratory at UMass Amherst. I did my thesis on the effects of strain variability on sauerkraut fermentation, a microbiological, chemical, and sensorial evaluation. Fermentation has been a method of preservation for many centuries. Fermented foods such as bread, pickles, and wines have distinct aromas, textures, and flavors due to the microorganisms present. Fermented foods are believed to help with digestion and have many microbes that are believed to be beneficial to the human microbiome. Fermentation is the process in which bacteria or yeast utilize sugars for energy. These foods are believed to contain microbes that are beneficial to the human gut microbiome. Spontaneous fermentation is a method of fermentation that uses the microbes naturally present in the food source rather than adding an additional ingredient such as a starter culture. In this case, the cabbage for the sauerkraut contains lactic acid bacteria that will utilize the sugars for energy and produce lactic acid, alcohols, and other byproducts, all of which affect the sensory characteristics of the sauerkraut, such as taste, texture, and aroma. For my project, I ran this experiment twice by creating 3 batches of sauerkraut samples. 2 contained starter cultures and 1 did not, meaning that it would ferment spontaneously. These batches of sauerkraut fermented over a period of 17 days in which the pH was monitored daily or every other day. Additionally, the sauerkraut samples were used to obtain standard plate counts by performing serial dilutions, and the brine of the sauerkraut was collected in order to test it for the concentration of organic acids. Once the sauerkraut was made, we were able to run a sensory test to determine whether the strain variability between samples affected the sensorial attributes. Lactic acid bacteria dominate fermented foods. In this study, 3 starter cultures were used. Starter cultures are very similar to instant yeast packets in the sense that they are a dried form of living microorganisms. The first trial used the cutting edge starter and the Caldwell starter culture, while the second trial used the cutting edge starter culture and the body ecology starter culture. The common strains within all 3 of these included Lactobacillus plantarum and Leuconostoc strains. These were all believed to dominate the experimental samples when doing plate counts. In the first trial, the pH of the samples decreased below 4 by day 3 of fermentation. We can see that the spontaneous experimental sample lags behind the 2 samples using starter cultures. Fortunately, all of these samples were able to drop below 4 by day 3 of fermentation, and this drop is crucial to the safety of the food and also contributes to the flavor. Similarly, the pH dropped below 4 by day 3 of fermentation in the second trial. However, starter culture 3, body ecology, decreased more rapidly than the other experimental samples. We can also see that there's a slight increase in the pH on day 7 due to some slight mold growth in the experimental samples using starter cultures, and this is most likely due to oxygen exposure when measuring the pH and removing samples from the fermentation vessel. In order to quantify the number of bacteria, I had to perform serial dilutions in order to obtain what is known as a standard plate count. I took the samples of sauerkraut and diluted them in order to plate them onto 3 different types of media. Tryptic soy agar, which is known as TSA, which is a general type of media used to grow many different types of bacteria. Demand rugosa sharp agar, known as MRS agar, is a selective media for lactic acid bacteria. Additionally, MRS with added sucrose was used to aid in the growth of Leuconostoc. The results shown were difficult to interpret as the bacterial counts fluctuated over a period of 17 days. The relative abundance of these samples generally seemed to decrease over time, however, the results were inconclusive. Similarly to the first trial, the 3 medias TSA, MRS, and MRS and sucrose were used, and the results were also fairly inconclusive as the standard plate counts fluctuated over the fermentation period. Once the sauerkraut fermentation completed, my team and I were able to conduct a sensory test known as projective mapping. Projective mapping is a sensory study that generally uses a 2D plane on which participants group the samples by similarities or dissimilarities. In this case, we were able to create this test using a database called CompuSense, in which participants could perform the projective mapping on a tablet. We used both experimental samples that I had created and commercial samples from the store to obtain the similarity data. The participants tried 8 different samples that were labeled with blinding codes and were able to group the samples on an online interface. One of the benefits of using this type of sensory study is that the panel does not need to be trained and the sample size can be fairly small. I was able to recruit 42 participants over the span of 5 days. The goal was to determine whether the addition of starter culture affected the sensory attributes of the sauerkraut. Here we can see some examples of ramekins with 20 grams of sauerkraut sample. Each of these ramekins had a blinding code posted to the front of the ramekin, and the tray that we gave to each participant had 8 samples in a randomized order so that participants would not be eating the samples in the same order. This image shows the interface that the participants were viewing during the test. They were able to drag the sample numbers onto the plane and group them however they chose to do so. What we found was that the majority of the participants could distinguish the experimental samples from the commercial samples based on the crunchiness and color of the experimental sauerkrauts. Since the data showed that the experimental samples were grouped together while the commercial samples were grouped farther apart from them, we were unable to confirm that the starter cultures affected the sensorial attributes of the sauerkraut. The overall results found were that the pH was able to drop as expected, allowing for a normal fermentation rate. Since the lactic acid bacteria standard plate counts were inconclusive, qPCR could be used in the future to quantify the DNA. Lastly, the sensory study concluded that the experimental samples were similar to one another and fairly different from the commercial samples, meaning that there has not been confirmation that the starter cultures affect the sensorial attributes. In the future, we will use high-pressure liquid chromatography to confirm the amount of organic acids present in the sauerkraut samples, as well as using cell pellets that have been saved throughout the process for 16S sequencing. This will help to determine the overall microbiome of the sauerkraut. And eventually, we would like to isolate the bacteria from the sauerkraut samples in order to create our own starter culture to then use in future sauerkraut samples. I would like to thank David Sella and his laboratory that have been incredibly helpful to me throughout the experimental process, as well as my research committee, including David Sella, Amanda Kinchla for product development and food safety, Alyssa Nolden for sensory, and Asha Rani for laboratory management. And lastly, Bridget Farah, a UMass alum that had started this project in the past and helped me recreate it this year.